Welcome back to our lecture series on structural characterization of materials. I am Professor Ashutosh Tiwari from the Department of Material Science and Engineering at the University of Utah. In the last lecture, we saw that the resolution of a scanning electron microscope is not determined by electron optics alone. Instead, SEM resolution is governed by three interacting factors. The first is probe size, which is controlled by electron optics. The second is pixel size, which is set by the scanning and sampling. We spent considerable time understanding how an operator can control these two parameters through lens settings, aperture, working distance, magnification, and scan settings. The third factor, however, is fundamentally different. It is the interaction volume, which depends on what actually happens when the electrons enter the specimen. In today's lecture, we will focus on this third limit and examine the physics of beam-specimen interactions, how electrons scatter, lose energy, and generate signals inside matter. To understand why beam-specimen interactions matter, we need to shift perspective. Once electrons leave the objective lens, the microscope no longer controls them. From that point on, the physics of electron-matter interaction takes over. Inside a solid, electrons do not travel in straight lines. Instead, they undergo multiple scattering events as they interact with atomic nuclei and electron clouds. Each of these interactions can produce a different detectable signal. Secondary electrons, backscattered electrons, X-rays, or Auger electrons, each carrying different information about the material. An important consequence of these interactions is that the electron beam spreads as it penetrates the specimen. This means that even a perfectly focused probe at the surface becomes broader inside the material, directly affecting resolution, contrast, and analytical accuracy. To understand beam-specimen interaction, we need to look at the problem at the atomic scale. The incident beam consists of high-energy electrons, which are negatively charged particles. As these electrons enter a solid, they experience the electric fields associated with atoms in the specimen. There are two primary sources of these fields. First, there is the strong positive Coulomb field of the atomic nucleus, where the positive charge is highly concentrated. Second, there is the more spatially distributed negative charge of the atomic electrons surrounding the nucleus. Interactions with these two regions of the atom occur in very different ways and lead to fundamentally different scattering behavior. This distinction forms the basis for understanding how electrons are deflected, how they lose energy, and how different signals are generated in the SEM. Let us first consider elastic scattering. In elastic scattering, the incident electron is deflected from its original path, but its kinetic energy remains essentially unchanged. This interaction is dominated by the strong Coulomb field of the atomic nucleus, where the positive charge is highly concentrated. Because the nucleus is so massive compared to the electron, momentum is transferred primarily as a change in direction rather than energy. As electrons undergo multiple elastic scattering events, the beam spreads laterally as it penetrates the specimen. This beam broadening increases the effective probe size below the surface, even if the incident probe is extremely small. After several large-angle elastic scattering events, some electrons can be redirected back out of the specimen. These electrons are detected as backscattered electrons or BSE. Elastic scattering, therefore, plays a central role in determining interaction volume size, beam spreading, and compositional contrast in SEM. The scattering cross-section, denoted as Q, is equivalent to the probability that an incident electron is elastically scattered by an angle greater than some minimum angle phi 0. This expression here shows how elastic scattering depends on atomic number, electron energy, and scattering angle. For SEM imaging, the most important result is the scaling behavior. The elastic scattering probability increases approximately with z square and decreases with increasing beam energy as 1 by E square. This scaling directly explains why heavier elements produce stronger backscattered electron signals and why increasing beam energy reduces elastic scattering and increases penetration depth. The distance an electron travels between successive elastic scattering events is known as the elastic mean free path. Physically, the elastic mean free path tells us how frequently electrons are deflected as they move through a material. When elastic scattering occurs often, the mean free path is short and the beam spreads rapidly. The expression shown here relates the elastic mean free path to material properties and the elastic scattering cross-section. You do not need to focus on the numerical details. What matters is the physical dependence. Materials with higher atomic density or higher atomic number cause electrons to scatter elastically more frequently, resulting in a shorter mean free path. Increasing the beam energy reduces elastic scattering and increases the mean free path. The elastic mean free path therefore plays a key role in determining how the interaction volume develops inside the specimen. In inelastic scattering, incident electrons transfer part of their energy to the specimen. During this process, the electron loses energy to the material rather than simply changing direction. This energy transfer can excite atomic electrons, leading to the emission of secondary electrons from very near the surface. If inner shell electrons are ionized, the subsequent relaxation of atom produces characteristic X-rays. In some cases, relaxation occurs through the emission of Auger electrons, which are extremely surface sensitive. Energy transfer can also excite lattice vibrations or phonons, contributing to local heating rather than useful imaging signals. Inelastic scattering therefore governs what signals are generated during electron-matter interactions rather than how the electron beam propagates through the specimen. It is useful at this point to clearly separate elastic and inelastic scattering side by side. In elastic scattering, the electron primarily changes direction without losing energy. This interaction is dominated by atomic nucleus and is responsible for beam spreading and the generation of backscattered electrons. As a result, elastic scattering plays a key role in limiting spatial resolution. In contrast, in inelastic scattering, the electron loses energy to the specimen. These interactions are dominated by the atomic electron cloud and lead to the generation of secondary electrons, characteristic X-rays, and Auger electrons. Inelastic scattering therefore controls image contrast and enables compositional and chemical analysis. Keeping this distinction in mind will help you understand the origin and meaning of all SEM signals discussed in the remainder of the chapter. As electrons enter the specimen, they do not travel in a straight line. Instead, repeated elastic and inelastic scattering events cause the beam to spread with depth. The region over which these interactions occur is known as the interaction volume, typically having a teardrop-like shape beneath the surface. The size and shape of this interaction volume depend strongly on the beam energy and on the properties of the material, particularly atomic number and density. Higher beam energy generally increases penetration depth, while higher atomic number and density tend to confine the interaction volume closer to the surface. An important point to remember is that all SEM signals originate from within this interaction volume, but not from the same depth or region. Understanding this is essential for interpreting image contrast and analytical signals. Although all SEM signals originate from the interaction volume, they do not come from the same depth within that volume. Secondary electrons are generated very close to the surface, typically within the top 1 to 5 nanometers, which is why SEM imaging provides excellent surface sensitivity and high spatial resolution. Background electrons originate from deeper regions, ranging from tens to hundreds of nanometers, depending on beam energy and material properties. As a result, BSE images carry compositional information but with lower spatial resolution than SE images. X-rays used for EDEX analysis are generated over even larger depth, often exceeding into the micron range, which limits spatial resolution for compositional analysis. All the electrons, like secondary electrons, originate from the extreme surface within only 1 to 2 nanometers. The key point is that each detector effectively samples a different interaction volume and therefore has a different intrinsic resolution. Backscattered electrons are generated through elastic scattering of the incident electron beam by atomic nuclei in the specimen. Because elastic scattering involves little or no energy loss, these electrons retain most of their original energy. However, they undergo large angle deflections, often reversing direction and escaping back out of the specimen. An important characteristic of backscattered electron is that their yield increases monotonically with atomic number. Heavier elements scatter electrons more strongly, leading to a higher probability that electrons are backscattered. This strong and monotonic dependence on atomic number is what gives rise to Z contrast in backscattered electron images, allowing compositional variations to be visualized directly. To quantify backscattered electrons, we define the backscatter coefficient denoted by eta. The backscatter coefficient is simply the ratio of the number of backscattered electrons to the total number of electrons incident on the specimen. In practice, this ratio can also be expressed using measured electrical currents, the backscatter current divided by the beam current. This parameter provides a direct quantitative measure of how strongly a material elastically scatters electrons back out of its surface. This plot shows the dependence of backscatter coefficient on atomic number. The key point is that eta increases monotonically with Z, meaning heavier elements backscatter more electrons due to stronger elastic scattering. Because a measurable signal varies systematically with a specimen property, this monotonic relationship directly produces atomic number contrast or Z contrast in BSE imaging. Regions with higher Z appear brighter. An important practical detail is that the shape of eta versus Z is steep at low atomic numbers and flattens at high atomic numbers. As a result, BSE contrast is strong for light elements but becomes weaker when distinguishing between heavy elements. For practical calculations and simulations, empirical expressions are often used to estimate the backscatter coefficient as a function of atomic number. This polynomial expression captures the observed behavior of eta over a wide range of atomic numbers and is commonly used in contrast modeling and quantitative interpretation. We now turn to secondary electrons, which provide a very different kind of contrast in a scanning electron microscope. Secondary electrons are generated through inelastic scattering of the incident electron beam with atoms in the specimen. In this process, energy is transferred to loosely bound electrons, allowing them to escape from the surface. Because of the way they are generated, secondary electrons have very low energies, typically less than about 50 electron volts. As a result, only electrons produced very close to the surface, within roughly 1 to 5 nanometers, are able to escape. This extremely shallow escape depth makes secondary electrons highly surface sensitive. Rather than emphasizing composition, they primarily provide topographic contrast, revealing fine surface features, edges, and surface roughness. To quantify secondary electron emission, we define the secondary electron coefficient denoted by delta. This coefficient is simply the ratio of number of secondary electrons emitted to the number of electrons incident on the specimen. Experimentally, it can also be expressed as the ratio of the measured secondary electrons current to the beam current. Because secondary electrons yield depends strongly on the surface geometry and local fields, secondary electron imaging is a dominant mode for high-resolution surface imaging in SEM.